Welcome to this video on epistasis and Bombay phenotype in humans. I will define the epistasis, explain the experiment used, findings and their interpretation, and finally I will explain Bombay phenotype in humans. In the end, I will also present a query to test retention of information during this lecture. So watch till the end. I will present briefly the overview of epistasis. According to Mendel, genes controlling different traits assort independently during inheritance. This is also known as the law of independent assortment, or in other words, you can say different genes do not interact. However, Lucien Cugnot in 1902, based on his two-trait mouse model, demonstrated gene-gene interaction. Later, William Bateson in 1909 introduced the term epistasis to explain gene-gene interaction. In the next slide, I will explain the two-trait mouse model conducted by Lucien Cugnot, which led to the discovery of epistasis. I will now explain the mouse model of two-trait cross that led to the discovery of epistasis. There are two genes, capital A, capital A, capital C, that regulate agouti mouse fur color or coat color, which is a dominant phenotype. In this experiment, pure breed agouti mice was crossed with a pure breed albino recessive mouse, and in F1 generation only agouti color mice were produced having dihybrid genotype capital A, small a, capital C, small c. This supports the law of dominance. However, the question was, what is the fate of recessive trait? To address this question, in the next experiment, the F1 dihybrid mice were self-crossed. A cross between dihybrid mice in P2 generation resulted in the formation of 9 agouti, 3 black and 4 albinos. This ratio 9:3:4 is in contrast with what Mendel reported 9:3:3:1. The reason for this will be explained in the next slide using Punnett square method. To use Punnett square method, it is important to write gamete genotypes from a dihybrid such as capital A, small a, capital C, small c. The first gene A is a hybrid, so split capital A from small a. The second gene C is also hybrid, so each point again is split into two, capital C, small c, capital C, small c, and put the combination here. And therefore a dihybrid such as capital A, small a, capital C, small c can produce only 4 types of gamete genotypes. So using this information, we will use Punnett square method to explain the ratio 9:3:4. So let us explain the ratio 9:3:4 using Punnett square method. This is male mouse forming 4 types of gametes. We have seen that, and 4 types from this female mouse. 4 times 4 is 16. Make 16 boxes and put the combination of these genotype and interpret the results. Wherever you have at least one capital C, one capital A, that will be agouti. So these are 9 agouti. Wherever you have at least one capital C and both small a's, that will be black. So there are 3 black, and ideally this should be one only albino, but there are 4 albinos. So what is the source of 3 additional? So these are 3 additional. If you notice here, there is a dominant gene, but this is being inhibited by small c, small c. So this is an example which clearly shows interaction between the gene or inhibition of a dominant gene by a recessive gene. This is called as epistasis. So epistasis is defined as inhibition of a dominant gene by a recessive genes present at a different locus. I would like to present now Bombay phenotype as an example of epistasis in human. There is a gene called H gene which is present at chromosome 19. This gene produces H-antigen which is subsequently converted into A, B, AB or O blood group depending on the presence of allelic genotype on chromosome 9. However, when H gene is recessive, H antigen is not present and therefore blood group A, B or AB is not produced by the corresponding allelic genotype. They all produce blood groups similar to O blood group. This type of O blood group, which is produced due to absence of H antigen, is known as Bombay phenotype. So in summary, when H gene is dominant, it produces blood group A, B, AB or O by corresponding allelic genotype present on chromosome 9. This type of O blood group is produced by the presence of H-antigen. However, when H gene is recessive, H-antigen is absent, and as a result of that, blood group similar to O blood group is produced by all allelic genotype present on chromosome 9. So this type of blood group which is produced due to absence of H-antigen is known as Bombay phenotype. There is a query: what is the difference between the O blood group and the Bombay phenotype blood group? The answer is O blood group is due to homozygous recessivity of the O allele, whereas Bombay phenotype is due to homozygous recessivity of the H gene, which masks ABO gene expression. Thank you very much for watching this video. I have uploaded all the small video clips to help you to revise the concept and experiment related to Mendel's inheritance, and that will help you to understand the whole concept. Please like it, share it, and subscribe it. Until next time, you take care. Bye bye.